Thank you very much for letting me present a little bit about what Model AD is. And Model AD really means Model Organisms Development and Evaluation for Late Onset Alzheimer's Disease. So a big mouthful, but really very much like what Marcelo was talking about. We have Alzheimer's disease, which has been talked about as being the 21st century plague, and it really is a major disease because in the US alone it has 5.8 million people that are affected by it presently, and it costs $290 billion per year in care cost. So that means 1 in every 5 Medicare dollars is going to AD care. In addition, if we don't find some kind of way to stem this or slow the progression of the disease by 2050, there will be 14 million affected in the United States alone at $1.1 trillion, i.e. 1 out of every 3 Medicare dollars as it stands now, would go for Alzheimer's disease and Alzheimer's disease-related disorders, dementia care. So Alzheimer's disease is a progressive neurological disorder, and it presents first with a loss of cognitive ability. For those of you that don't know, they have the pathological hallmarks in this human brain are amyloid protein deposits, and what looks like here on these black arrowheads is dead neurons. Those are neurofibular tangles, or tau, hyperphosphorylated tau. You can see here that an AD brain, this is a human brain, has a lot more crevices and a lot broader versus the control brain, and that's because there is a loss of neurons, and it is the loss of neurons that causes that really is what's the cause of the dementia. But what's causing the loss of neurons? And so what we also see in the pathology is there are these cells that are in there that become reactive, indicative of inflammation, which are microglia and astrocytes. And so in order to investigate some of these diseases, these really complex human diseases, there's lots of different approaches one can take, particularly with human samples, looking at the clinic, imaging, tissues, and cell models. But what we're doing is looking at animal models, and that's the one we're choosing because here, by studying animal models of a human disease, you can test drugs in those models before you start testing them on humans, which would be a good idea, we think. So this is what Model AD is doing now. In order to think about getting a model, an animal model of a human disease, you have to sort of consider what you have to do. And it's likely that we're going to have to humanize some of these mouse genes, and particularly those that are key to AD, like an amyloid gene or the tau gene, to overcome some inherent differences between the human and the mouse. We could talk about that later if you were interested in that. In addition, there's multiple pathologies that you see in humans, and it may be that not all of those pathologies will be seen in one mouse model, so you're going to have more than one mouse model. Finally, there's also genetic effects, there's aging, there's the environment, all of these things have to be considered, and then there is a PC to Mac deficiency here. But nevertheless, a mouse strain background and the very complex genetics that all of us have. We are not inbred, which makes it very hard to work on us. In addition, very few of our human participants will donate their brain for, while they're living, for analysis. So what we can find out, though, is stuff about the genetics. And we know that a lot of the animal models that have been going on before were based on genetic mutations in these 3 genes. It doesn't really matter what they are, but they do cause Alzheimer's disease, very early onset, and pretty much 100%. They are rare mutations, but the risk of getting Alzheimer's disease is very, very high. However, that comprises only about 1% to 2% of all the individuals that have Alzheimer's disease. So what we do know that there are a lot of other genetic factors that have much lower risk conferred when you have these genetic variants, but they can be very common. Some of these are up to 40% of the population have those. Now, these do not cause Alzheimer's themselves, but in combination then, could be factoring into the onset and progression of the disease. So what's Model AD doing? We want to prioritize these different variants for late onset Alzheimer's disease so that we can put them into an animal model and create these new genetic mice with the new CRISPR mechanisms that you might have read about. But we're using some old-fashioned recombination methods as well so that we put these genetic mutations into the mice. But what we have to do is select from this large number of possible genetic mutations, and we have to do that. We have to prioritize that. And so we look at how many studies do you see these variants coming up in Alzheimer's disease? Is there a predicted effect on the function in Alzheimer's disease? Is there a differential expression of those genes in Alzheimer's disease? And how close are the mouse and the human genes so that we can manipulate them without too much trauma to the mouse? And this is are there non-coding variant effects, which we won't need to go into. But once we have prioritized these, and you can see that these are in a number of different kinds of pathways, we can then begin to characterize those mouse models once we have put them into the mice and see which ones of those, which kinds of characteristics will come back and look like the humans. And what I mean to say, and this we've seen in the previous test, is we're going to take some of these mice, new mouse models, we're going to look at RNA sequencing and the different omics. Does any of those look like any of the people that have Alzheimer's disease? If we look neuropathologically, do any of these mice that we have here, do any of those look like some of the people with Alzheimer's disease? And again, in brain imaging, can you do this? So these are a lot of different kinds of technologies, and so you need to have a lot of different expertise. Once we find a mouse, the red mouse that looks like the red person, the red Alzheimer's person, we can use different drugs in those particular mice, see if they are going to be protective against the disease, and those then would be candidates for use in those red Alzheimer's disease population. All of this is based on open science. All of the models that we generate are freely available to anybody without any legal restrictions. There's no patents, there's no licensing, there's no nothing. Everything is out there. All the data that we get is also open sourced, and you can get it through the websites. So what are our deliverables? We want to do 50 new mouse models of AD. We're about halfway there now and a little bit halfway through the grant. We've already got a mouse that has human A beta. We're working on the human tau now. And when we then put these other variants onto this platform mouse, we're going to test them in various diets and various kinds of environments to see if that makes a difference in speeding up Alzheimer's disease. The whole goal, again, is to share all of these mice models and the data that we get from them with other people to try their good ideas out so that we can get rid of this disease fast. Here at UCI, we've also then leveraged this whole thing by bringing in multiple other people into the group here. Ian Smith, for example, and Andy Obanas has come in, and a number of other people, Katrine Whiteson, doing different things on these mouse models, trying to characterize and trying to look at different factors. And we've actually then been able to bring in addition to the core grant, an additional $2.8 million in supplemental support in the 2 years that we've had this grant. So that's good, and that gets everybody then working together and makes it very exciting. So what does it take for this? This is our core team, so to speak. Frank LaFerla is co-director with me on this whole thing, but we really need a synergy of these different expertise across these different team members, and they all have different capabilities. Institutional infrastructure, like a computer. Yeah. And then also communication is very, very important. And what's really important in all of this then is mutual respect of the team members and their capabilities, diversity of opinions and diversity of individuals coming in with different ideas, transparency, no hiding, everything is open, and as Marcelo once said, fearlessness. So you just get out there and do it. Okay, so this is our team here. It's great fun. This is the rest of the team that do a lot of the work here. But we also work in conjunction with other labs, Indiana University, Jackson Laboratories, and SAGE, which are also in this Model AD consortium. We have actually bi-monthly meetings with them, weekly meetings with our NIH program officer, and we have an annual external advisory board of very distinguished individuals. All of these people then are trying to work out so that we can get rid of this Alzheimer's disease and be able to sustain our health care. So thank you, and if you have any questions, I'd be happy to answer them.
Thank you, Dr. Tenner. Are there questions for Dr. Tenner?
Yes.
Can you say a little more about how you go about open sourcing and sharing your data and models?
Okay, yeah. So the models that we make, the mouse models, we ship them to a facility called Jackson Laboratories. That's a commercial laboratory, and they breed them up and check them to make sure that they are the right background, and then they sell them. So they're not free, but there's no legal restrictions. You don't have to pay a license fee to UCI or to anybody, and commercial groups can have it. So they're academics can have them. That's the one. That's for the mice, and for the data. I guess I didn't put the website up here, but if you go on to MODELAD, it will give you directions to the AMP-AD portal, and that's the Accelerated Medicines Programs for Alzheimer's Disease that is sponsored by NIA. And as soon as when we characterize our mice, all the RNA-seq data is uploaded, all of the immunohistochemical data is uploaded, the behavioral data is uploaded. LTP, that's all going to be uploaded and in that portal. Now, it might not be easy for a 2-year-old to do, but, you can get to it.
A little bit more than that. Okay, we have to move on. Thank you so much, Dr. Tenner.